Okay, uh, welcome back. Uh, can you hear me?
Yes, sir.
Yes, sir.
Sir.
Clear and loud?
Yes, sir.
Yes, sir.
Okay, then let's start. So in the last class, we actually uh, we've been discussing this transpositions, types of transpositions, isomers in bacteria, and transpositions in bacteria again. Now today we'll discuss a specific topic, uh, transposition, hybrid dysgenesis that occurs due to transposition of P elements, again, the transposition in Drosophila. So we'll see how this transposition of P element causes hybrid dysgenesis. So before we go to the hybrid dysgenesis mechanism, let's try to understand what the hybrid dysgenesis is. Now, to begin with, the P elements are transposable elements that were discovered in Drosophila as the causative agents of genetic traits called hybrid dysgenesis. Okay. Now, the hybrid dysgenesis syndrome in Drosophila is marked by temperature-dependent sterility, elevated mutation rates, and increased chromosomal realignments and recombination. Now, all these mutations and realignment that cause actually sterility in the progeny, uh, and this is temperature dependent. Means that if temperature is elevated, then they no longer is capable of producing offsprings. So overall, we find a lot of morphological changes due to mutations, and so far these reproduction is concerned, they produce in F1 generation uh, the sterile offspring. Okay, now let's make it more clear. Uh, Suppose these are parents. This is female and this is male Drosophila melanogaster. Uh, Now, when they cross in certain cytotype, we'll discuss and explain this cytotype. But for this moment, just try to understand that in a certain type of cytotype, mm, distribution in male and female actually causes this, or produces sterile offspring or progeny. Okay, but again, you see the distribution of P element, whether it is present in male, it actually determines whether the progeny will be sterile or not. In this case, you see when uh, female is having P cytotype and male is having M cytotype, mm, then the uh, offsprings progeny produced due to the crossing of these parents, it doesn't become sterile. It is normal, and it can again produce progeny. Okay, so it is normal flight. But when here you see when female is M cytotype, but the male is P cytotype. Here male was M cytotype. Uh, Here the case was reverse. Here the female is M cytotype, male is P cytotype, and when the cross occurs between them, they produce the offspring. That's okay, but further it cannot produce uh, the offsprings. So it is that is why it is called sterile progeny. Okay, and it is also known as dysgenic progeny. So along with these, you may see or people may find some other mutations also, phenotypic mutations. Okay, now let's explain what are these M cytotype, P cytotype, when they're when the female will be P cytotype, when the male will call P cytotype or M cytotype. Now let's look into that. Okay, uh, as I said, this P element is transposon. It is actually class 2 transposon, uh, and it moves by cut and paste mechanism. So in the earlier class, we discussed about these two mechanisms. One is copy paste mechanism, another is cut paste mechanism. Now this P element, this transposon uh, present in Drosophila, actually class 2 type transposon, and it follows cut and paste mechanism for transposition. Now, if you look at the P element or transposon at molecular level, then you will find that it comprises 4 exons and 3 introns, okay, like this. So the gene of this transposon contains 1, 2, 3, 4, 4 exons, and in between 3 introns, 1, 2, and 3, 3 introns. Now, this is one stop codon. The star indicates the stop codon is present in this intron. Okay, third intron. Well, now, uh, you know, when the nascent mRNA is processed, one of the important processing of mRNA is splicing of introns in eukaryotes. Prokaryotic genes do not possess the intron sequence, but eukaryotic genes possess intron sequence, uh, non-translating sequences in genes. Therefore, when the mRNA is produced from the gene, the nascent RNA, it needs to go for processing to produce mature mRNA, which will eventually be translated in cytoplasm. Uh, but this processing occurs in nucleus itself. So, as Drosophila is eukaryotic organism, and its genes processes introns. So these introns need to be, spliced out. Uh, but interestingly, there occurs differential splicing. It means uh, in some cells, uh, uh, this both 3 introns get spliced off, uh, but in some cells, only these 2 introns get spliced off, but the third intron remains, and the the mRNA after splicing, it go actually it comes out of the nucleus and moves into cytoplasm along with this third intron, and that there in cytoplasm, this ribosome tries to translate the whole mRNA, but well, when it comes to this intronic portion, it encounters this uh, stop codon, and it cannot translate further. Therefore, the exon 4 is not translated when these kind of this kind of uh, immature or differentially spliced mRNA moves to the cytoplasm. Now, mm, uh, there are a lot of examples of this kind of differential splicing, and it gives rise different uh, types of proteins from a single gene. This is also one mechanism to produce variation in proteins. Okay. Now, the complete splicing of introns produces the transposis enzyme. Okay. Now, mm, when these three introns are spliced off, and from exon 1 and exon 2, exon 3, exon 4 are spliced together, and when the translation occurs, it produces a longer, you know, mm, uh, it produces longer peptide. It's because it doesn't stop at the stop codon, and it produces a bigger protein, 87 kilodalton protein. But when this intron remains, then the gene is translated uh, up to this region only, up to you can say this is exon 3 up to exon 3 and a little more, and this gives actually a smaller peptide, because this exon 4 cannot be, this part cannot be translated, therefore it gives a smaller peptide, 66 kilodalton protein. And now I'd say that both the proteins are functional. Uh, 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 it may seem like that, it may appear like that, that as because it was not fully translated, and the protein which is produced from the truncated protein which is produced from these exons, uh, uh, the smaller peptide is produced from this, uh, I mean, mRNA. Uh, it may be inactive protein, but but here this is not actually inactive protein. The smaller protein has also role to play. Interesting. Uh, uh, you will find it interesting how it plays a role of repression of transposition. Okay. Now, again, uh, uh, as I said, that complete splicing of introns produces the transposis enzyme, while alternative partial splicing of intron 1 and 2, leaving in only intron 3, encodes a repressor molecule, the P element repressor molecule. It's a smaller peptide. Then, what does this transposes uh, causes? What does this? What is the function of the transposes? The transposes enzyme, which recognizes 31 base pair terminal inverted repeats of P element. You know that every transposon actually possesses inverted repeat at both the ends of the transposon, whether they flanks, this inverted repeat flanks the central part of the transposon. Here in P element, this inverted repeat is 30 base pair, 31 base pair long. And these 31 base pair is actually recognized by the transposes. And after that, it catalyzes P element excision and reinsertion. Okay, so this longer protein, which is the transposes, so it causes the transposition of the P element by this mechanism in brief. Okay. In the absence of P element repressor, so when this P element repressor is not there, so transposers can act. But when P element repressor is present, if somehow this repressor is present, then it inhibits the function of transposes, and transposes cannot actually then recognize or eventually cannot catalyze the p element excision and reinsertion. Okay, so in the absence of p element, the p element represent the p elements can proliferate throughout the genome. In absence, it can proliferate throughout the genome. It can be inserted throughout the genome. But the problem is this insertion causes disruption of genes. So it is responsible. So it inserts, it may insert, I mean, arbitrarily, and in some cases, it can insert itself within a gene, and then it actually disrupts the gene, and therefore the genes get inactivated. So if it is a very important gene, and if it controls one phenotypic character, then what will happen? This phenotypic character will be, appear abnormally. And if it is a very important gene, it may also kill the organism and killing the protein. Okay, so depends what kind of genes are getting affected. Okay, so now we talked about the cytotype, P cytotype and M cytotype. Now, how it is related with this cytotype is related with hybrid dysgenesis. Okay. Now, the dependence of hybrid dysgenesis on the sexual orientation of a cross shows that the cytoplasm is important as well as the P factor. So, along with the P factor, along with the transposon, the cytoplasm of the cell is also important to this hybrid dysgenesis to occur. Uh, the contribution of cytoplasm is described as cytotype. Okay. Now, a line of flies containing P elements, okay, they have a P cytotype, while a line of flies lacking P elements has M cytotype. So, you can call the cell not possessing the P transposon actually having N cytotype, and the cell having P elements in the chromosome in the genome actually possess C cytotype, P cytotype. Okay. Now, it has been found that hybrid dysgenesis occurs only when chromosomes containing P factors find themselves in M cytotype. Okay, so let me explain this. Uh, uh, so hybrid dysgenesis occurs only when chromosomes containing P factors find themselves in M cytotype. Then how can it happen? Okay, because P elements having the chromosome, the cell having P elements, it is actually having P cytotype, cytoplasm. Now, how come uh, this uh, P factors find them in M cytoplasm? Because when P factor is uh, present in the chromosome, automatically the cell cytoplasm is P cytoplasm. The answer to this question is suppose uh, this P element uh, is transferred to a cell having M cytoplasm from outside, then this P factor can find themselves in M cytoplasm, right? So how then how does happen naturally? The answer to this question is in during fertilization, If a sperm is having a P element in their genome, uh, then this sperm can donate this P factor as because sperm genome actually get transferred to the ovum into the ovum during fertilization. So the female ovum could be M cytotype. Uh, this ovum may not have. I mean, uh, suppose the one is not having the P element, therefore the ovum cytoplasm is P cytotype. Uh, so when the sperm enters the ovum with P element, then it happens that P factors find themselves in M cytotype. And when this kind of fertilization occurs, then the male parent has P elements and the female parent doesn't. So that is in this case, the male parent has P element, okay, but the female parent doesn't. That is why female is M cytotype. And in that case only, these P factors actually this transposon get transposed. Okay.
স্যার
Yes.
স্যার কোন একটা অর্গানিজমে যদি পি ফ্যাক্টরটা থাকে ওইটা কি মানে সার্ভাইভ করবে না?
ওই পি ফ্যাক্টরটা যদি থাকে সে সার্ভাইভ করবে করবে কারণ আমরা দেখবো মলিকুলার মেকানিজম, কিন্তু আমি এখন একটু বলে দিচ্ছি করবে কারণ পি ফ্যাক্টরটা ওখানে ইনঅ্যাক্টিভ কন্ডিশনে থাকে। ওকে, আমি আমি এটা এক্সপ্লেইন করব যে কেন কেন ইনঅ্যাক্টিভ কন্ডিশনে। ওকে
স্যার ওই আগের স্লাইডটাতে লেখা ছিল যদি পি ফ্যাক্টরটা মানে সাপ্রেসরটা যদি লেখ থাকে ইন অ্যাবসেন্স অফ দ্য পি এলিমেন্টস রিপ্রেসর দ্য পি এলিমেন্ট ক্যান প্রলিফারেট থ্রু আউট দ্য জিনোম ডিসরাপ্টিং মেনি জিনস এন্ড কিলিং দ্য প্রোজেনি।
রাইট, এটাই তো। So this is the last paragraph. In the absence of P element repressor, the P elements can proliferate throughout the genome, disrupting the many genes, killing the progeny. Okay.
তাহলে স্যার রিপ্রেসরটা থাকা মানে তো পি এলিমেন্ট মানে যদি অ্যাবসেন্স অফ পি এলিমেন্ট রিপ্রেসর তার মানে তো পি এলিমেন্টটা থাকবে।
P element আছে, P element এর repressorটা নেই, কিন্তু P element is there. Okay. যদি in absence of repressor P element is there, so P elements can proliferate throughout the genome. ঠিক আছে, এবার question is if P element is there without the repressor, then P element will proliferate and the cell or the organism will get killed. Okay, now you see that it is written over here progeny. Okay, so progeny means in the next generation, but the P element, if the P element is present in absence of repressor, then the organism will survive. So let's explain it. This is the point which I am going to explain. Okay. So we have already discussed this P cytotype and the M cytotype. Hybrid dysgenesis occur only when chromosomes containing P factors find themselves in M cytotype. Okay, that is when the make. So up to this, is it clear?
Yes, sir.
Yes, sir.
Okay, just follow the next slides. Okay. Okay. Now, here I have put one question again. Why hybrid dysgenesis manifests when crossing P-strain males with M-strain females, and not when crossing P-strain females uh, with M-strain males. Okay, so try to get this question. Before that, let me jump this slide for the moment to we just try to focus in this paragraph or this one. Activation of P element is tissue specific. Okay, so I'm explaining the question you have asked. You have asked a nice question. So uh, the activation of P element uh, is tissue specific. So it occurs only in germline. Okay, but P elements are transcribed in both germline and somatic tissues, though they express in both in germline as well as in somatic tissues. Uh, their you know expression is different in germline and somatic tissue. Okay, and we will see that. So we must remember this. Yes, in somatic tissues, only the first two introns are excised, creating a coding region of ORF0, ORF1, and ORF2. Okay, ORF3 is missing, and this causes the production of 66 kilodalton repressor. Okay, now going back to yours, so you just remember in germline tissues, both the all the introns get removed, and the ORF tree also get expressed. I mean translated, and this so therefore in germline, all four open reading frames are actually connected. Uh, mm, they get connected to get the mRNA, nature mRNA, and when it is translated, it produces 67 kilodalton transposase enzyme. That is why it gets activated in germline, because we get the bigger protein transposase, and when transposase is there, then only it will be transposed and will get activated. Okay, now going back here, so uh, here uh, in the absence of P element repressor, the P elements can proliferate. Now you see that if the P element is present in the somatic cell, okay, the P element is there in the somatic cell, but it will produce only repressor, because in somatic cell the splicing of the third intron doesn't occur. So repressor will be there, and even though the P elements are there, they will not move to other places. Okay, and will not disturb the genes. That is why, though in spite of having these P elements, they will survive. Okay, so only in progeny, because to produce progeny, they have to have gametes, to produce gametes, male and female both, and gametes are germ cells. And when this P element goes to the germ cell, then their expression gets changed. There actually they produce the you know full transposase engine. So after that, when this gamete actually fertilize the ovum, okay, the cross occurs between the you know, the parents, having different cytokines, then actually this phenomenon occurs. Let's again go to the molecular basis of the hybrid dysgenesis. Okay, now I've already explained this tissue specific expression of the element, and now in somatic tissues, this is also I have explained. Now this is what happens in if this mm, this is short RNA, it's a long RNA. So intron 1, intron 2, this blue parts are introns, and so with introns, it is being transcribed. Now, what happens in somatic cells? In somatic cells, this third intron remains, and eventually, as because there is a codon, translation don't occur beyond this point, and a smaller peptide, 66 kilodalton peptide, which is a repressor, it is produced. Now, in germline mRNA, here you see this loose intron, third intron has been spliced, and the whole reading frame is actually translated. Therefore, in germline, this 87 kilodalton bigger protein is produced, which is transposes, and therefore P elements get activated. Okay. Now, so whenever YF3 is spliced to the preceding reading plane, the P element becomes active. This is a crucial regulatory event, and usually it occurs only in germline. So you know that now. What is responsible for the tissue specific splicing? Now you can ask why it is so. Why this in somatic cells it same gene produce smaller peptide, but in germline it produces bigger transposes and gang. The answer to this question is in somatic cells, a protein uh, is present that binds to sequences in axon tree to prevent splicing of the last intron. So due to the presence of this protein, which prevent the splicing of the third axon, actually this intron remains. The absence of this protein in germline cells allows splicing to generate the mRNA that codes the transpose. Here, now the transposes binds to 10 base pair sequences that are adjacent to the 31 base pair inverted repeats. This transposition occurs by non-replicative carbon-based mechanism resembling the PN10 transposon. It contributes to hybrid dysgenesis in two ways. One way is it disrupts the genes if it is get inserted into the gene, so it disrupts the genes. And another way is that so where from it actually leaves, the site where from it comes out, there actually a gap actually is produced. Like this, you know, when it is removed from the donor site, you see a gap is produced. So this is also called chromosomal breakage, DNA breakage. But sometimes, you know, this breakage is actually filled up through, you know, recombination. But If it is not filled up, then this will remain. So in two ways, when it is moving to the target site, if target site is having an important gene, then you see the gene is getting disrupted. So it is getting bifurcated. So it may no longer remain active. It may no longer produce active protein. So this is one inactivating gene. Another one is that producing breakage at the origin where from it is coming out. Okay. Now, uh, the 66 kilodalton repressor protein is provided as a maternal factor in the egg. Now, you know this. Uh, why maternal cytoplasm, I mean ovum cytoplasm is having this 66 kilodalton repressor protein. Because you know when in the egg is fertilized,